Shruti, you are on live.
Good evening to one and all present here. Vaccines are the most cost effective public health intervention we have, protecting not just individuals but entire communities. This powerful reminder reflects the importance of immunization, awareness, and action in preventive healthcare. With this thought, I warmly welcome you all to today's insightful webinar, Session 4 of IndiVax 2026 Future Frontiers in Vaccinology, part of the state level vaccination webinar series organized by the Indian Medical Association Medical Students Network, Tamil Nadu State Branch, supported by the IMA Junior Doctors Network, Tamil Nadu. A good beginning sets the tone for a meaningful journey. To formally welcome our gathering and set the stage for today's session, I invite Aditi Balan, Academic Subcommittee Member, IMA-MSN TNSP, to deliver the welcome address. Over to you, Aditi.
Good evening, everyone. It gives me immense pleasure to welcome you all to IndiVax 26 Session 4 Future Frontiers in Vaccinology. I would like to begin by respectfully acknowledging the presence of our esteemed patrons Dr. Sridhar Sir, Dr. Sengutavan Sir, Dr. Ravi Kumar Sir, Dr. Tyagarajan Sir, Dr. Tirivya Mohan Sir, and Dr. Solomon Jha Sir. Your continued guidance and encouragement serve as a strong foundation for all our efforts. We are also privileged to have with us our respected state team, Dr. Annamalai Sir, Dr. Jamuna Rani Ma'am, Dr. Kannan Rajendran Sir, Dr. Arunagiri G Sir, Dr. Ajay Karthik K Sir, Dr. Bala Hariharan S Sir, and Dr. Harinam Vijay Sir, and also our organizing team whose dedication has made this event possible. IndiVax 26 is more than just a gathering. It represents a shared vision, a vision where knowledge meets purpose and where we, as future healthcare professionals, come together to understand and strengthen the role of immunization in protecting communities. We are truly fortunate to be joined by an exceptional speaker today, Dr. J.D. Jahapriya Ma'am, Associate Professor of Microbiology in Srinivasan Medical College. Ma'am's expertise and experience promise to provide us with valuable perspectives and inspire meaningful discussions. As we come together for this event, it reflects our collective responsibility to promote awareness, improve accessibility, and contribute to advancements in vaccination and preventive healthcare. I extend my heartfelt thanks to all our speakers, faculty members, organizers, and participants for being part of this initiative. Once again, I warmly welcome you all. I hope this session proves to be insightful, engaging, and impactful for each one of us.
Thank you, Aditi, for that warm and gracious welcome. The right introduction is not just about credentials, but about connecting us to the mind behind the knowledge. To introduce our esteemed speaker for the day, I now invite Harisha, Academic Subcommittee Member, IMA-MSN TNSP. Over to you, Harisha.
Good evening, everyone. It is my pleasure to introduce our esteemed speaker for today's guest. Is a highly accomplished clinical microbiologist with over a decade of experience in the field. She completed her MD in microbiology from the Tamil Nadu Dr. NGR Medical University and has been actively involved in both academic and clinical practice. Currently serving as an associate professor at Srinivasan Medical College and Hospital. Dr. Jayapriya Ma'am has been actively involved in NABL and NABH accreditation processes, molecular diagnostics, including RT-PCR hospital infection control programs. Ma'am has also contributed to several research studies and has presented her work at national and international conferences. A passionate educator and researcher, she is currently working on a developing microbiology textbook for undergraduate students and PG aspirants, reflecting her dedication to academic excellence. We are truly honored to have her with us today for sharing the invaluable insights. Let us warmly welcome Dr. J.D. Jahapriya ma'am. Thank you.
Thank you.
Thank you for that wonderful introduction, Harisha. Prevention is not just better than cure, it is the foundation of a healthier society. It is now my privilege to welcome our speaker, Dr. Jahapriya Ma'am, Associate Professor, Department of Microbiology, Srinivasan Medical College and Hospital, Trichy. We are truly honored to have you with us today, ma'am, and the stage is now yours.
Thank you. Thank you, Shruti. First of all, very good evening, pleasant good evening.
And I thank the entire IMA team, especially the student network, for organizing such a wonderful and insightful, useful webinar series for the purpose of other fellow colleagues. So definitely, I must congratulate all of you for this wonderful work, and I wish you to continue this throughout your lifetime career. Okay. So, and I thank the patrons, and I thank the state team and other organizing team members. So, without much any delay, so let us jump into the topic, that is future frontiers in vaccinology. So, for this, for understanding this particular vaccine series, you must know a little bit basics. So, in this session, I'm going to highlight what are the innovative methods, what is the research, importance of the research in the vaccine department, vaccine development, and also to highlight the next generation vaccines when comparing with the traditional vaccines. So, what are the things have been changed? What are the advantages of the next generation vaccine? And what are the technologies which have been improved, and why it is needed, and to prevent the millions of deaths, especially this vaccine coverage which happened in the COVID season, which we all are going to come across in this 1 hour session. And the last but not the least, what is the role of your people like young medical scholars? So what is your role in this vaccine department? And last is what is our role towards the public health? So these are all my highlights. So by the end of this session, you will be able to how the vaccines are developed. So I'll be explaining both traditional and also the newer generation vaccines, and I will explain the entire mechanism of each classification of the vaccines. And let us see what are the challenges in the emerging diseases, and what are the hurdles in vaccine development, and how to implement from the lab side to community side, and to understand the medical students, especially what is the role of yourself in future generations. Okay. So with this question, I want to start my session. So why vaccines matter? So after hearing the 3-4 previous sessions, I hope you all would answer. So you can unmute and answer. Why vaccines matter? Can anybody answer? Just quickly, for 30 seconds. Can somebody unmute and answer? I want some 1 or 2 points.
Ma'am, it helps in prevention of diseases, ma'am.
Excellent.
Ma'am,
Any
it
other
provides
point?
herd immunity.
Yes, very good, excellent. Even in unvaccinated individuals also, we can cover them through the some part of immunized people. We can cover the unvaccinated individuals. Very good point. Any other point?
Ma'am, it works on the concept of prevention is better than cure, ma'am.
Okay, so I think Shruti explained that same concept. Prevention of many diseases. Prevention is better than cure. Same point. One more thing, which is important nowadays, money. So it helps in reducing the healthcare cost. Okay, by taking, so that is prevention, right? If we take vaccine as a prevention, the cost will be reduced. So that is the reason vaccine matters to the individual, to the community, and also to the future generation. Already we have seen smallpox has been eradicated by effective vaccination, and ongoing polio control is being achieved now, even in the measles also. So that is why vaccine matters. And again, one eye-opening question: how we achieved successful rate in case of this COVID outbreak within a short time frame by maintaining the safety and efficacy? Can you answer this question? Okay. By the end of my last slide is the same. You will be able to answer after this session. I hope the question is clear. Okay. So I hope my objective and vision is clear. So what is the history of vaccine? The first vaccine was invented by Jenner. So Jenner learned இப்போ current இருக்க COVID Johnson and Johnson vaccine வரைக்கும், which is the key tool which drives the vaccine department itself, that is research. So research was the one which made all the scientists to be awakened, to make them not to sleep, and also to discover and lots of vaccine trials. Okay, so that is what from the Jenner learned Johnson and Johnson vaccine varikume research. Only by research people achieved all the vaccines. Okay, so I don't want to go into history much. And another question: dash is the active ingredient of the vaccine which stimulates an immune response against a specific disease causing organism? Only one answer.
Antigen.
Excellent. So that is antigen. What is antigen? What is antigen? Can anybody answer?
Basically
When
recognizes a foreign sub and triggers an
Enapa?
immune response. Basically recognizes a foreign body and it triggers the immune response.
Yes. So antigen is the one which is going to stimulate and trigger our immune response. Do you think the whole antigen will trigger?
Only a part, man.
What is
The
that part
epitope.
called? That
Epitope.
part is called epitope. So epitope. What is epitope now? Now the definition is going to be a little bit changed. Only one word you have to add. So what is epitope? Epitope is the
Part of the antigen.
part
Crossing,
of the antigen, specific molecular region of the antigen. Okay, so that is what epitope. It is the antigenic determinant of the whole antigen, which is going to trigger the immune response by binding with the B cell or B cell receptor or T cell receptor. Okay, so epitope plus antibody. This binding nadanda mattuda immune response will get stimulated. So based on this same principle is applied in the vaccine also. Vaccine has to be developed in format of epitope. Okay, so epitope மாதிரி தான் vaccine இருக்கணும். But it has to lose its antigenicity property, but it should have the immunogenicity property. So an antigen should have two property. One is antigenicity property, another one is immunogenicity property. Is that clear? Antigenicity means able to produce enough amount of antibodies. Immunogenicity means immune response should be triggered in a enough amount so that the protection rate will be high. So that's what the same principle is applied in the vaccine. So how we can classify now vaccine as traditional and newer? Why the shift has happened from traditional to newer? That is what the topic today. So what are the traditional vaccines include? Live attenuated, inactivated, which means killed. Okay, whole organism can be killed and used, or only a particular unit of the organism, that is called subunit. Only the toxin can be used, only the protein can be used, only the polysaccharide can be used, or by recombining with any other inert particles like yeast recombinant therapy can be used, or even outer membrane protein can be used. So similar, these are all the subunit. A part of the antigens can be used as a vaccine. Idella traditional. So we started from live, killed, and then subunit. Why the newer generation vaccines have developed? What are the newer generation vaccines? We are going to use virus as a vector to deliver the vaccine. That is called virus vaccine, virus vector vaccines. Instead of using virus-like particles can be used, like chemical. Because here virus is involved, right? So obviously there are some side effects will be there. So which I'll explain in later slides. Okay. So we can use the whole virus, or we can use virus-like particles like chemicals instead of using the viruses, and nucleic acid, that is RNA or DNA. So these are all the next gen vaccines. Are you clear with the types? Shall we move on to the next slide? Yes. So how live attenuated vaccines work out? So what is mean by live attenuated? We are going to use a live virus, but we are going to make some changes in case of attenuation. Attenuation means weakening. So weakening of the live harmful virus, weakened vaccine genome is delivered. Once a weakened vaccine genome is delivered, it is going to trigger the immune response. So what is a major disadvantage? Still the virus can be alive. Okay, that is why it is contraindicated in immunocompromised patients. The immunocompromised patients, what will happen? This avirulent can become virulent. Okay, this reversion can happen. So it will be having some disadvantages. But the advantage is it can trigger the immune response very, very strongly. That is one advantages of live attenuated vaccines. What are the examples of live vaccines? MMR and 2-pox, chickenpox. Okay, and smallpox. So what are the inactivated vaccines? What is the role of inactivated vaccines? We are going to kill the harmful viral genome, destroy. We are going to destroy. That is called kill by either chemical method or heat method. Okay, inactivated means which the virus can't replicate, which means we can use those inactivated viruses in case of immunocompromised patients also. So safety, 100% safety in case of all the patients, okay, irrespective of their immune status. But what is the disadvantage? Still, though it is inactivated, immune response might be minimal or moderate. That is why it requires multiple doses. So are you clear with the live and inactivated vaccines? So that is why subunit vaccines develop now. So what are the purpose of subunit vaccine? Instead of giving whole virus, instead of giving killed virus, recombinant or polysaccharide or conjugate or outer membrane proteins or proteins alone can be used. But what is the disadvantage? By giving only little bit part of a antigen, it has to get combined with adjuvant to enhance its immunogenicity. So this adjuvant preparation அவங்களுக்கு ஒரு struggle இருந்தது for the researchers. So without the adjuvant, the effect will be minimal. So adjuvant enhancer should be added with the target protein or vaccine. Is that clear? So these are all the subunit vaccine. Again, subunit vaccine can't produce stronger immune response, so it requires repeated multiple doses. Okay. So moved on to the toxoid. Same toxins, conversion of toxin to harmless is called toxoid. Along with the toxoid, you have to give adjuvant enhancers. Mostly exotoxins will be used rather than endotoxins. Is that clear? So inactivated toxins will produce immune response. Except live vaccines, மற்ற எல்லா வேக்சின்ஸுமே ஆன்டிபாடி ரெஸ்பான்ஸ் தான் இன்டியூஸ் பண்ணும். In case of live vaccines, you can see both cellular and antibody. There are two types of antibody response. One is cell mediated, another one is antibody mediated. Live vaccines are capable of producing, they are potent stimulator of both innate and adaptive immune response, unlike inactivated or subunit. I hope we all are clear with the traditional vaccines. So having this basics, I have put up some advantages, disadvantages, examples all together in one slide, which we discussed already. So what is translational vaccinology? Bringing the vaccine from the lab side to the public side. That is called translational vaccinology. It's a methodology. Okay. So once from the lab side, the antigen is extracted and it's put into the preclinical stage where animals are tested by giving the vaccines, whether it is sufficient dose is there, by what route it should be given. So all those testing will happen in the first animals. And then it is put onto the clinical trial where all the 4 phases it has to pass through. Initially, small groups will be involved to look for the safety, and the larger group to look for the immune response and the dose. And phase 3 include larger populations where the effectiveness, whether in their serum, enough amount of antibody response are produced or not. So to look for the response, serological antibody titer, that is called the effectiveness, and also to look for the side effects. And last is phase 4, that is after getting approval from the authorities, it has to be given into the circulation, and also we have to monitor for the side effects. Okay, so after the clinical trial, it should go to regulatory approval stage, and then manufacturing distribution in a large scale amount, and then public health implementation monitoring. So from bench to the bedside, this entire process is called translational vaccinology. Okay, so what is reverse vaccinology? How it differs from traditional vaccines? So we all just saw that, right? What are the traditional vaccines? First, we grow the pathogen, we weaken or we kill it, we extract only a particular unit, and we have to make vaccine. This is called traditional vaccines. So how it differs from reverse vaccine? Reverse vaccine means first step itself studying the genes. Okay, then you assess and predict the antigen, anticipate பண்ணி நீங்க antigens இதுவா இருக்கும்னு தெரிஞ்சுக்கிட்டு you are going to design vaccine, and then delivery, manufacturing, distributed to the public. So what is the advantage of traditional and reverse? Can you tell me one advantage from this particular slide? It is there, it is obvious. Will you go with traditional or reverse from this slide? Which is easier?
Ma'am, traditional is easier.
Traditional is easier. Okay. Any other different opinions? Which is safety?
Reverse, ma'am.
Reverse is safety. Yes, for of course safety purpose, this reverse vaccinology, that is what we faced during COVID outbreak. How was that possible? In a shorter period, we didn't even reach after 3rd wave, right? So how is that possible? The safety. Okay. So 100, even n number of 100 times the safety about the reverse vaccinology, because we can't anticipate. Here, in case of traditional, we won't know the outcome, right? Only we have made vaccine, but we don't know the outcome. Here, we know the genes, we know the methods, we know the antigens predict பண்ண முடிஞ்சிச்சு. So we know what kind of antibodies, what kind of immune response will be developed in a patient or in a public. So that is why reverse vaccinology have been evolved. Is that clear? So we'll move on to what are the next gen vaccines. What is reverse vaccinology? Is otherwise known as modern vaccines. Modern vaccines are also called as reverse vaccinology. I hope this will be a newer term for you. That's why I put that word. And what are the next gen vaccines? Which we discussed already. Either we can use viral vector, virus as a whole vector to carry the vaccines, or we can use viral-like particles, VLP, that is all the nanoparticles, protein nanoparticles like chemical substances, or we can go for nucleic acid, DNA or mRNA. I think we are clear with the next gen vaccines. Modern vaccines using advanced technology, another advantage is faster to design and produce, then more targeted immune response, because we know what kind of antigen we are going to give. So this is more targeted and personalized. That is why 100% safety. Okay, is that clear? Whereas opposite, in contrast to the traditional vaccines. Okay. So again, I have put up all the examples in one slide, which we'll discuss. So first we'll see one by one. Viral vector vaccine. So what are the viruses which can be used for carrying the vaccines? Most common is adenovirus or adeno-associated virus. And why particularly in the virus mattu choose pandrom? Any points? Why adenovirus alone? There are many viruses are there. Adeno-associated virus, even retroviruses, gamma group, especially gamma group, from pox viruses. There are viruses, but why this particular viruses? Anybody? What is our role ultimately? Viruses and what is the definition of virus? Basics always starts with the basic. Viruses are obligate intracellular, right? So same concept. If suppose this is a virus, is having the vaccine. Okay, virus kulla ipo vaccine, that is nothing but viral vector vaccine. This has to be injected, right? And this viruses are obligate intracellular. Oru cellukku innoru cellukku the same vaccine has to be transferred. So what is that process called? Gene transfer. Okay, gene transfer, you know all the methods of gene transfer, right? Transduction, transformation, conjugation. So this particular adenovirus has high transduction efficiency. Okay, that's why cell to cell transfer rate will be very, very high. That is why we can achieve the response very immediately. Immune response can be triggered instantly. Response will be higher when compared with other viruses, number one. Another one is, that is what? Potency, speed, potency, and scalable. Production. Again, production is very, very good in case of this one. Stability. It's a DNA virus, right? So stability. Any other points?
Ma'am, adenovirus usually doesn't integrate into the host cell.
Yes, very good. Zero or minimum genomic integration. Okay, so there is less risk of infection. That is what's saying. Genome integration rate will be very, very minimal. So by all these reasons, adenovirus has been used. The same mechanism I have put it in this picture. Here, target virus protein is inserted into the virus, harmless virus genetic material. There are some other virus material also, even adenovirus or a genome also will be there. Okay, along with that genome, this vaccine genome has to get integrated and it is given. Okay, and what is the disadvantages? What Shruti said is genomic integration. So in case of the patient might have got previously adenoviral infection, so in such condition rarely it can cause reactogenicity. So obviously they will be having side effects, right? So instead of using, that is why Johnson & Johnson vaccine in the previous slide what I showed, which was prepared for COVID, was withdrawn because of this viral vector vaccine. Okay, and to avoid that, viral-like particles were started in use. Okay, what is viral-like particle abina? First, we have to study the virus. First, number one. And what virus can be used? Ada padichittu, they will genomic study பண்ணுவாங்க. What are the proteomics and genomics and the departmentல கேள்விப்பட்டிருப்பீங்க. Study of proteins, study of genomics, and virus has to put into all the computer-aided techniques like proteomics, genomics to study all the structural, non-structural protein which is highly immunogenic and which has to go for latency and which will cause carcinogenic and all. So once it is studied, once the coding and decoding அதெல்லாம் பண்ணதுக்கு அப்புறம், and it has to study which particle can be used. That is called virus-like particle. And chimeric VLP with foreign peptide, or can it be given alone, or it has to be added with any other immunostimulatory signals, which I will be showing in the DNA. Okay, it has to be added with some other lipid particles, so that cells are made up of phospholipid membrane, right? So on the lipid membrane, which is very, very thick, to cross that membrane, this structure has to be made. Okay, so cations எல்லாம் add பண்ணுவாங்க too, because negatively charged cells are nucleus are made up of negatively charged, so adding cations, all those work will be happen. Thus that is called foreign peptide. இந்த மாடிஃபிகேஷன்ஸ்லாம் பண்ணதுக்கு அப்புறம் ப்ரிப்பேர் பண்றது தான் VLP. Even the VLP has made a drastic change in the vaccine research development. See, what are the VLPs can be which has been used அப்படின்னா bacterial cells. Another one is, rather than virus cell, like how we saw adenovirus, instead of using adenovirus, even bacterial cells also were used, and insects, yeast cells, mammalian cells, plant cell. This plant cell where ரொம்ப ரிசர்ச் பண்ணது வந்து பிளான்ட் செல்ஸ்ல தான். ஓகே, பிகாஸ் ஆஃப் லோ காஸ்ட், தட் இஸ் வை. அண்ட் வாட் ஆர் த வேக்சின்ஸ் வேர் ப்ரொடியூஸ்ட் பை VLP டெக்னாலஜின்னு பார்த்தீங்கன்னா HPV, தட் இஸ் கார்டாசில் அண்ட் செர்வாரிக்ஸ், அண்ட் ஹெப்படைட்டிஸ் வேக்சின்ஸ், ரீகாம்பிவேக்ஸ். இது எல்லாமே இட் ஷோட் சக்சஸ்ஃபுல் ரேட் இன் கேஸ் ஆஃப் VLP. ஓகே, ஸோ தட் இஸ் ஈவன் SARS-CoV-2 ப்ரோட்டீன், தட் இஸ் கோவிஃபென்ஸ் அப்படின்னு சொன்னாங்க. ஓகே, பட் கோவிஷீல்ட், ஃபைசரும் மாடர்னாவும் நியூக்ளிக் ஆசிட் வேக்சின்ஸ். தேர் வேர் ரிசர்ச், many researchers were done in the COVID, because not only SARS-CoV-2 the outbreak, previously there was MERS and MERS-CoV irukkalaya, so apparinde coronavirus was there, so there were many researchers being done. It didn't sudden or knee jerk vanda madri 2020 la the outbreak na kadaya. So there were many researchers done, so that is why this mRNA vaccine was possible by that time. Okay, so that is the VLP. Yeah, this was the example. Hepatitis B and HPV, Cervarix, Gardasil, which is still in use, right? And in 2025, even first approved vaccine for chikungunya was developed. Okay, is that clear? So moving on to the nuclear-based vaccine. Since here it looks complex and costly, right? Because we have to develop lot of foreign peptides, we have to manufacture a lot of the things, and stability issues are there, manufacturing issues are there, scalability, and large, whatever we have to manufacture, it should be in a large population, large amount of quantities also. So all those issues are there in the virus and viral like particles. Viral vector vaccines, we have to maintain the cells, that is why major hurdle for virus vector. Viral like particle looks costlier than the virus, because it has to be made of all chemicals. So that is why newer vaccines like nucleic acid based vaccines develop, that is DNA and RNA. I hope so far we all are clear with the vaccines. Shall we proceed, or any doubts? Shall we proceed with the pDNA vaccines and mRNA vaccines?
Yes, ma'am.
Yes. So what is pDNA? What is mean by P? I have put P here. Can anybody answer? It is there in the slide.
Plasmid DNA.
Yeah. What is plasmid? So you should know. If you know plasmid, you also should know about the chromosomes. What is plasmid and what is chromosome?
Ma'am, plasmid is the DNA material located outside the nucleus.
Yeah. So this is nothing but extra chromosomal substance. This is where the nucleic part is there, DNA part is there. Okay. So why we chose plasmid? Why not chromosome?
Ma'am, it confers antibiotic resistance.
Sorry, guys, we are handling a little bit of network down issues. Ma'am will be back with us soon.
Is anyone contacted ma'am Shruti Lavinash? Ma'am, contact பண்ணீங்களா?
சார் நான் பண்ணல சார்.
ஓகேடா.
Okay, is it visible now?
Yes, ma'am.
Yes, okay. Sorry for the interruption. So why plasmids are being chosen than the chromosome? Because it is having the self-replicating property. Okay. It doesn't require any chromosome, like only chromosome can be able to divide only if the cell is present. Okay. In the presence of cell, chromosome divides like 1, 2 or 2, 4 or adu madri. This is irrespective of the cell. That is why plasmid is being chosen, number one. Another one is they can act as a better carrier than chromosome. So plasmid, like 2-layer circular membrane kuda or vaccine component can be inserted and it can be given as a vaccine. So that is why DNA plasmid is being chosen. So idu kuda nariya immunostimulatory conjugates are needed because since it's a nucleic acid based vaccine, it has to enter into the host cell or nucleus. It shouldn't get diluted in the cytoplasm. So it should be enough, it should be having the enough stability property to cross the cell membrane layer, nuclear membrane layer, and to reach the nuclear part. That is why it has to be conjugated with some other immunostimulatory components like viral capsid can be used, or nanoparticles can be used, or cationic conjugate, as it has to interact with the negative nuclear component. It has to be made with the cationic conjugate, and golden beads, liposomes, or even empty bacterial capsules can be used. So along with all this, putting all this together, so plasmid has to be separated, plasmid has to be chosen, and purification and conjugation, and then insertion. Okay, so these are the steps of plasmid DNA vaccine preparation. So by what method it has to be delivered? There are many methods are there: intradermal, intramuscular, biolistic, depot, mucosal, electroporation methods, sonoporation method. By many methods it can be given. Okay, along with some adjuvants to enhance its immunostimulatory component. Okay, so this is how plasmid vaccines differ from the conventional one. So conventional, once it is injected, it has to enter into the cell, endolysosomal fusion happen and then proteins will be released. Proteins will be broken down by the proteasomes, and protein recognition will happen by the MHC molecule, and MHC molecule will showcase a particular protein what is injected via vaccine to the CD4 T cell via T cell receptors, and immune response will be there. So more amount of antibody immune response will be predominant in case of conventional vaccines. In case of newer vaccines, these are all the gene gun, tattooing method, injection method, edible vaccines are DNA vaccines. So it enters into the nucleus directly. That is why the DNA vaccines are the advantage. So once the DNA is entered, DNA has to convert into mRNA. What is that process called? Transcription. So first transcription nadaku, then mRNA gets translated into producing more antigenic proteins. So that proteins will be processed by the proteasome. It will be cleaved and more protein peptides will be produced. Okay. So this particular protein peptides act in intracellularly and also extracellularly. Once it is considered intracellularly, CD8 T cell will get triggered. If it is considered extracellularly, CD4 T cells will be triggered. So, which means both class 2 MHC and class 1 MHC pathway will get activated, by which thereby antibody response also will be there, T cell response also will be there. So, cell mediated immunity or humoral immunity response will be there. So, that is the advantage of newer vaccines when compared with the conventional vaccines. I hope I'm clear with the plasmid DNA vaccines. These are all the different routes by which we can achieve the immune response. So if it is given by intramuscular, myocytes will be triggered. Gene gun, Langerhans cells. Tattoo gun, dendritic cells. By oral route, M cells, intestinal M cells will be triggered. Is that clear? So the same picture I have put example for a tumor antigen. Okay. So what is mRNA vaccine? This is the last part of the session. What is mRNA vaccine? So mRNA has a unique role. Okay, you know what is mRNA? It's a single strand structure. It has 2 ends, 5' and 3'. So each component has its own role. Okay, 5' if mRNA is given as a component carrying the vaccine, it should not get degraded, because when comparing with the DNA, mRNA is very, very weak, right? It's single stranded. DNA is double stranded, it will be more stable. So it has to be prevented by the degradation, first one, and improve stability. So that is why the 5 cap, that is the role of 5 cap, okay, 5 dash cap. And translation efficiency, enhances the efficiency and reduces the immune response optimization. It helps in optimization. The other end is translatory region. That is, function of this is facilitate the ribosome recruitment. More amount of ribosomes are recruited, more amount of proteins will be secreted. So that is the reason for that is the role of 5-UTR. And what is this ORF, which is very, very important? ORF component encodes the target protein, which is carried by the as a vaccine. Okay, and same, idellame prevents the maintains the stability part. This is the structure of mRNA, and we are going to insert the vaccine target protein into this mRNA, and it is delivered. And what are the delivery methods? Same, lipid method, polymer based, peptide based. It can be added or adjuvanted with all these components. Okay. So the classical example is spike protein. COVID-19 mRNA spike protein has to be inserted in the mRNA. Okay. And same, these are all the various particles which can be used to deliver the mRNA vaccines. And yes, once the first step is vaccine injected intramuscularly, and it delivers the mRNA into the host cells, that is muscle cell or dendritic cell. Next is mRNA. mRNA role in it has to get translated into proteins. So that is the next step. Next step is translation. இதெல்லாம் எங்க நடக்குதுன்னா, it remains in cytoplasm. Unlike DNA vaccines, it happens on everything happens in the nucleus, whereas mRNA happens in the cytoplasm. As Shruti said previously, genomic integration possibility happen in the DNA vaccines, whereas it is avoided, it is neglected in case of mRNA vaccines, because this happens entirely in the cytoplasm, where the ribosomes translate the mRNA into proteins. Then antigen processing happens. Both, it can trigger both class 1, class 2 pathway, by which both the T cells can get activated, and both cell mediated immunity இருக்கும், antibody response also will be there. Okay, so immune activation, antibody response. This is the mechanism of mRNA vaccine. And this came and swiped the entire traditional vaccines. Okay, the same mechanism I have put up in a picture format, and same mRNA vaccines. So what are the advantages of mRNA vaccines? Why we talk about mRNA? Because of safety, no live pathogen is used first, no nuclear entry, so negligible risk of genomic integration, and good efficacy rate, strong humoral and cell mediated, both arms of immune response will be potent and stimulated, and rapid development, fast design, sequence based design, no need of using pathogen culture and all. Whereas pDNA cell, it requires cell culture, okay, to maintain that plasmid, whereas here it is completely in vitro. That is why no pathogen culture is required, and platform flexibility is very, very good, easy adaptable, especially in case of emerging pathogens, and scalable manufacturing. What are the limitations? Since it is single stranded, it's highly unstable, it's very, very, very less stable, so it requires ultra cold chain storage. There is a one major limitations of the mRNA. And reactogenicity is very, very minimum. It also because of innate immune activation, not because of the vaccines. And it delivery depends on the LNP technology, okay, nanoparticle technology, and booster doses may required. And what are the basic differences between mRNA and DNA vaccines? Where the location of action happens, here it is in the cytoplasm, whereas DNA nucleus. Mechanism, first, once mRNA is inserted, what will happen? Ribosome translate the protein antigen. So first step is translation, whereas here the plasmid pDNA, first it has to undergo transcription and then translation. Okay, transcription to mRNA to translation to protein. So here everything happens in the cytoplasm, it happens in the nucleus, whereas the speed is ultra fast. Here it is slower, because you have to develop pDNA, first extraction of the plasmid, purify it, so lot of works are there. Stability, it is very, very, very less, whereas this advantage of pDNA is higher stability. Okay, Genome delivery is lipid nanoparticles, whereas here lot of methods are there: gene gun, IM, tattooing, any oral. Genome integration risk is none. Okay, that is why mRNA is being the game changer. Okay, whereas here minimal, and also theoretically it's possible, but practically minimal. Immune response very, very strong. Okay, here moderate. I hope I made you clear with the DNA and mRNA vaccines. Okay, so this is the steps of production of mRNA. First, mRNA design panano, upper synthesis and delivery, then preclinical testing, fill finishing, then clinical trial, and then approval. Okay. These are the timeline of the mRNA vaccine since 1961 learned. For 2020 COVID, that is 2 mRNA vaccines which protected the entire world after the third wave, that is Pfizer and Moderna. Okay, 2020. So after 2020 mRNA இல்லையான்னு கேட்டா no, it is there. So after 2020 அப்புறம் பாத்தீங்கன்னா, apart from the infectious reason like genetic diseases, cancer, infectious diseases இல்லாமோ like autoimmune diseases, allergic conditions. So இதுலெல்லாம் வந்து mRNA along with monoclonal antibody. Okay, in case of any genetic disease, autoimmune diseases, it's being applicable. Yes, they are the Nobel Prize winners. What they did, what made them to achieve the Nobel Prizes, they converted the unmodified mRNA uridine வந்து pseudo uridine change பண்ணாங்க. So this one modification made us to talk about mRNA even today. What happens? Why this conversion pseudo uridine? Because all the AU, GC in all bases are responsible for immune inflammatory response and immune activation. Whereas they made this uridine inactive. So what happens? Inflammatory response is stopped. 100% stop. So obviously, reduce the innate immune activation, reduces the natural immune response against the organism. Instead of that, translation was more and more protein antigens were released and more immune activation against the vaccine developed. So that is why they achieved the Nobel Prize for the successful mRNA vaccine platform. Okay, Carico and Weissman. We must be thankful for them. And what is the role of AI in vaccinology? Any points you have? It will be helpful in antigen selection, immunogen design, and epitope prediction. You can predict the epitope. So which part is the hotspot of the antigen? Okay, and vaccine targeting methods and adjuvant identification and immunomodulatory methods, techniques, and optimization of the codons in case of nucleic acid vaccines and vaccine design pipelines. Okay, so these are all the in all these methods, AI will be helpful in vaccinology department. Moving on to the last part of the session is emerging infectious diseases. So what is mean by emerging infectious diseases? Emerging means, what is mean by emerging? Emerging na இப்பதான் புதுசா increase in incidence newly. Okay, then any other point for emerging? What is mean by emerging? One is increase incidence. அப்புறம் any 2 points? Are you people there? What is
Yeah.
emerging infection?
Something
Okay.
new.
Something new, yes, newly identified. Apro, it should cause increase in incidence rate. Apro, because it is emerging disease, right? That is why. Next, another point. It should bring all our attention. Geographically, it should bring our attention. These are all the points for emerging infectious disease. So why we have to talk? What is the reason for emerging? திடீர்னு ஏன் வரலாம்? Or zoonotic spillover or any human migration. Okay, there is global travel and speed of dissemination, high mortality, rapid mutation. Any other points? Urbanization, modernization, apro, climate change, because the climate change will alter the vectors or the multiplication also. Vectors habits also will be அதுவே ஒரு separate topic. Climatic change வந்து it's a very different topic to talk about that. Okay, so இதெல்லாம் the reason for emerging infectious diseases. There are a lot of emerging infectious diseases like 2000ல இருந்து பார்த்துக்கிட்டீங்கனாலே, see here, SARS-CoV, Marburg, swine flu, Ebola, yellow fever, COVID, Lassa, Zika, MERS, chikungunya, West Nile, dengue and all. See the mortality rate and all for Marburg, Ebola and all. It's almost more than 70, 90 and all. So that is why we have to develop vaccines for and emerging infections. We should keep more amount of researchers should be done, and we have to be alert always, as we can't predict the emerging infections anytime, right? That is why. So what is pandemic preparedness? Why pandemic preparedness should be done? One is to be alert. Then what is maybe pandemic preparedness? Why first? Because of delayed lab confirmation. Lab confirm panni நம்ம பண்ணணும்ன்றது we can't expect all the time, because in case of resource limited areas lab confirmation might be delayed. Okay, any other points?
To reduce mortality rates, ma'am.
Yes, very good. To reduce mortality rate. Non-specific clinical manifestation. That's what I said. Ipa pathina summer, but why we are having dengue? Yesterday also a dengue positive. But this is what climate change will alter the vector habits. That is why we can't predict what in the seasonality ellame change aayichu. Okay, that is why we have to be prepared all the time, and limited surveillance system also. So these are all the reason why we have to be prepared. Okay, and another one is, what is our, this we discussed already, herd immunity, disease eradication, reduce mortality. Herd immunity means interrupt the chain of transmission and gives protection to the unvaccinated individuals through vaccinated individuals. One, upper control of epidemic and also the pandemic. Okay, and economic impact also can be reduced by giving vaccinations. In case of emerging infectious disease, நம்ம இந்த லைவ், கில்டு, சப்யூனிட், ஈவன் நியூக்ளிக் ஆசிட் வேக்சின்ஸ் என்னதான் டெவலப் பண்ணி வச்சிருந்தாலும், by what strategy it will be useful? Can we vaccinate the mass whole people? Vaccinate பண்ண முடியுமா? Yeah, it is quite impossible. In that condition, we can apply something called ring strategy. இது எனக்கே புதுசா இருந்தது. I did research. So அந்த டைம்ல what is ring strategy அப்படின்னா, first primary population who are all affected with the disease we have to cover. Okay, first we have to vaccinate the primary contact, means those who are having the disease, and then covering the secondary contact, people contact with the people who got affected, who got the infection. Though ivangale vaccinate pannu. Okay, it's not possible to vaccinate the entire contact, at least few from the primary contact. So this is called ring strategy. Okay, when is it possible? When is the ring strategy possible? In case of any sporadic condition. It's not possible in case of pandemic condition, right? Pandemic condition means they already crossed the stage 3. That is community spread vandrum. Community spread vandadukku appuram neenga vandu selective vaccinate panna mudiyadhu. So in case of sporadic diseases like ipa paathola Zika, Nipah and all, here and there are some cases would be recorded. And the condition this is strategy is applicable. Okay, so combining epidemiological surveillance along with the targeted immunization. Classical example is Ebola, Nipah and all. Okay, there's one newer strategy where you can apply in case of emerging infectious disease, especially sporadic conditions rather than outbreak. Okay, sporadic outbreak rather than pandemic, I mean. So the last but not the least, that is challenges in vaccination. So what are the challenges we face in case of vaccine development? First one is, I think these are all the previously discussed topic. The first one is storage point. Storage means where the vaccine should be stored. Cold chain environment la da store பண்ணிக்கணும். Okay, so that is the one. Difficult in case of low resource settings, like it's very, very difficult. Another one is manufacturing challenges. It requires some advanced technology. Okay, that's we discussed, right? Genomics, proteomics, AI assisted, இதெல்லாமே advanced technologies where இருந்துச்சுன்னா we can manufacture it நிறைய. That is one of the challenges. And cost is especially cost, expensive to produce, to develop, and accessibility. This should be equality arkano. Okay, equity in accessibility and distribution. And safety, long-term data. Yes, limited long-term safety data in case of newer technologies. So that is why one hurdle for vaccine development. Newer advanced AI vechadella romba minimal. Okay. Then distribution, which we discussed. Public acceptance, that is vaccine hesitancy because of misconversation. Especially இப்போ COVID mRNA பார்த்தீங்கனாலே ஒரு misconception தான். So public acceptance is very, very, very difficult. Only by giving mass awareness we can achieve that. Okay, viral variants and mutation we can't prevent because of the climate, because of the pollution. There are many reasons for the virus to undergo mutations and all. Okay, then regulatory and approval issues, it's very, very complex and time consuming. Approval process ரொம்ப time ஆகும், and emergency approval will be decided by the government authorities, and it should need to be balanced, speedy with the safety also. So அதெல்லாம் அவங்க கிட்ட இருக்க regulatory and approval issues எல்லாம். So these are all the challenges in case of vaccine development. The last component of the session is, what is your role in future? As a young medicos, what is your role in future? Can you tell or 5 points irka? I have 10 points to discuss. I want 1 or 2. Anything? Okay. First is precision and personalized vaccination. That is called tailored vaccination. We have moved from the uniform immunization schedule and ipa tailored ku shift aayachu because of the advancements. So targeted immunocompromised ku in the vaccines, pediatrics ku in the vaccines ku we have been tailoring the vaccine schedule. First, another one is rapid vaccine development platforms have been developed. Okay, like ipada sonno Pfizer and Moderna. Throughout the world they achieved the COVID vaccine coverage. Okay, and then expansion into non-infectious diseases. So what is the future? Infectious disease மட்டும் தான் future ஆன்னு கேட்டா no. What are the other diseases? Any chronic inflammatory disease இருக்கு, autoimmune diseases, allergic conditions, then cancer. Cancer is emerging one, right? So cancer, இது எல்லாத்துக்குமே mRNA technologies are applicable. Okay, strengthening public health and preventive medicines. Okay, then integration with the digital health and AI, research and academic opportunities, lot are there, and ethical regulatory components, clinical relevance and daily practices. Okay, so what are the myths and fact? Do you think mRNA vaccines use new technology? No, already hundreds of scientists have been working on mRNA vaccines for decades. So இது வந்து புதுசு கிடையாது. Okay, mRNA vaccines will alter your DNA? Definitely not, because it never enters into your nucleus of the cell. So there is no genomic integration. There is no risk of genomic integration. It will not alter your DNA. mRNA vaccine contain microchips? Definitely not. Okay, they may have only the encapsulated particles like LNP as a delivering vehicle. They are just inert. Okay, and it enhances the entry, that's all. And mRNA vaccines ingredients are dangerous? Definitely no. Okay, do not contain any metals, any preservatives, any blood or any live cells. Okay, they are cell-free technique, so it is full of safe. And will it give me the infection? Definitely no, because there are no live viruses used. Will it cause any female infertility? Currently no evidence. Theoretically might be possible. There are no clinical evidence. Okay, I think I covered the session. Again, coming back to the same question, how it is achieved in a short time period, COVID-19 vaccine. I think now you'll be able to answer.
Reverse vaccine technology, mama.
1 point. அப்புறம் technique. So advanced technology use பண்ணாங்க. அப்புறம் this is one important slide where I want you to think. இந்த 1 hour நீங்க listen பண்ணதை வச்சு இந்த 10 points. I want at least ஒரு 5 points.
Using mRNA.
Other reverse, advanced or newer generation, anything. Global collaboration. Okay, because it was pandemic, right? Pandemic in rapper, obviously every countries will talk about it, every countries will discuss. So global collaboration, unity. Next, any point, any other points? Huge funding. Since it's a pandemic, funding இருக்கணும் இல்லையா? I said last point, regulatory and approval authorities வந்து support ஒரு hurdle. So அந்த டைம்ல, COVID டைம்ல it will be, அந்த இதெல்லாம் கிடையாது, நீயா நானாலாம் இல்லாம எல்லாருமே they work together, so that unity and funding helped them. Next, this is one important point which might be new for you. Parallel trial phase. So what is parallel trial phase? We saw, right? Phase 1, phase 2, phase 3 clinical trial. இங்க mRNA வேக்சின்ல the 1 and 2nd phase கம்பைன் பண்ணிட்டாங்க. Okay, during preparation itself they gave the first is checking the safety, second is checking for the dose and immune response, the third is side effects and effectiveness இருக்கு இல்லையா. ஸோ ஃபர்ஸ்ட் அண்ட் செகண்ட் ஃபேஸை கம்பைன் பண்ணிட்டாங்க. திஸ் இஸ் கால் கம்பைன் ட்ரையல் ஃபேஸ் ஆர் பேரலல் ட்ரையல் ஃபேஸ். ஒன்லி பை தென் அவங்களால டைம் பீரியடை கம்மி பண்ண முடிஞ்சு தே கவர்ட் லாட் ஆஃப் மாஸ் பாப்புலேஷனை வேக்சினேட் பண்ண முடிஞ்சுது. இது ஒரு இம்பார்ட்டன்ட் ஸ்ட்ராட்டஜி தே அப்ளைட். ஓகே, தென் எனி அதர் பாயிண்ட்ஸ்? லார்ஜ் ஸ்கேல் ட்ரையல். ஓகே, லார்ஜ் ஸ்கேல் ட்ரையல் அட் அ டைம்ல நிறைய ட்ரையல்ஸ் பார்த்தாங்க. Practically it's impossible if we want to give a live vaccine or inactivated vaccines and all. So in case of that outbreak, large scale trial was used. Any other point? Mass production. Enada?
Already, as you had mentioned, previous research work would have been done.
Previous research oda data வந்து நிறைய இருந்துச்சு. So which supported the researchers during the COVID period to produce mass vaccination. Okay, and manufacturing, mass manufacturing production, then efficient distribution system because of the like governments, NGOs avangala help பண்ணாங்க for the easy dissemination and distribution. Okay, and then சேஃப்டி மானிட்டரிங், இதுதான் ரொம்ப ரொம்ப முக்கியம். வேக்சின் போட்டதுக்கு அப்புறம் யூ ஹாவ் டு கம் அண்ட் இன்ஃபார்ம் த ஹெல்த் கேர் டீம் ரிகார்டிங் த வேக்சினோட சைட் எஃபெக்ட்ஸ் இருக்கான்னு. ஸோ சேஃப்டி மானிட்டரிங், only by all these steps it was possible. Okay, so I think I finished the topic. Any doubts இருந்துச்சுன்னா நீங்க கேட்கலாம்.
Ma'am?
Yes.
Ma'am, so we've been discussing about reverse vaccinology techniques.
Yes.
Ma'am. So
Yes.
recently we have been reading many articles regarding vaccination to cancer. So what's your talk on that, ma'am? Like how is it works?
Same. I put one slide. I think for example tumor antigen ku போட்டிருந்தேன். Same. இதே தான் ரீசன். mRNA vaccine, mRNA கூட anti-cancer drug or monoclonal antibodies can be inserted, and it is same technology. We can even cover the cancer treatment also.
Ma'am, but there will be individual variations, ma'am. How can we tackle
that? Is what personalized. இப்போ எல்லாமே personalized. They will take their host or a DNA எடுத்து, they will read that, they'll open it by proteomic genomic technology, they'll open it, they'll check the HLA compatibility, and then they will give. ஈவன் இப்போ எப்படி பாக்டீரியாவுக்கு நீங்க ஏஎஸ்டி பாக்குறீங்க, சென்சிட்டிவா ரெசிஸ்டன்டான, அதே மாதிரி ஆன்டி கேன்சர் ட்ரக்ஸையும் வச்சு சேம் அந்த எலிசா வெல் மாதிரி ஒரு ஆன்டி கேன்சர் ட்ரக்கையும் தே வில் செக் ஃபார் த அடாப்டபிலிட்டி, அண்ட் தென் தே ஆர் tailoring the vaccines, they are personalized, they are giving it to the patient. Even now இது more advanced இப்போ இதுதான்.
Okay ma'am, thank you ma'am.
That is why proteomic genomics அதெல்லாம் vast department where we can earn a lot, we can learn a lot also. Any other doubt?
Ma'am, you had mentioned in that ring strategy that the first group of people which we have to concentrate on involves the primary population where those
ring strategy.
Yes, ma'am. Those who had contacted the disease. Ma'am, are we allowed to vaccinate people who are who have contacted the disease during the disease process itself, ma'am?
That is not possible in case of airborne infection and all. That is why Ebola is more spread through transmission, direct contact நால மட்டும்தான் வரும். They will select the target individual who are all eligible for getting vaccine. They will not consider the immunocompromised status. They may be considering the healthy individual whom they will go and contact with the patient. Only they will cover that particular group. That is what I told, selectively from the second ring and the first ring.
Okay, ma'am. Thank you.
Where we can't produce mass vaccination for, we can't expect the lab also to produce mass vaccination against the Ebola and all, because it will be useless. Since it is contact based and all, we can contain them very easily. Unlike COVID we couldn't even contain, right? It crossed from country to country, sea to sea. So that is why it's not possible in case of airborne the land. Direct contact transmission it will be possible.
Okay, ma'am.
Okay da. Any doubts? Any other doubts? Okay. So I thank the IMA team for this grateful opportunity. I think I made you all to be aware of this current vaccines and future vaccines and the previous vaccines. Anything else?
Ma'am, there has been a question in the chat box. Is there any fellowships for vaccinology, ma'am?
Any?
Fellowships for vaccinology, ma'am.
Fellowships for vaccinology. Fellowships for immunology is available. Immunology fellowship courses irukku. Only in particular institutions like AIMS irukku, CMC irukku, JIPMER I'm not sure. AIMS and CMC irukku. Immunology irukku. If you finish immunology itself, you can go and work in other biotechnology people and you can learn a lot. There are many hopes and there are many chances are there, opportunities are there in that technology department, biotechnology and all.
Okay, ma'am. Thank you, ma'am. Thank you.
Thank you. Can I leave or is there anything to wind up?
Yes, ma'am. We'll just wind up the meeting, ma'am.
Sure.
Thank you, ma'am, for that incredibly enlightening session. Ma'am, your session gave us deeper understanding on the newer perspectives and the nuances and technologies for the future and for the present, ma'am.
Thank you.
Gratitude turns a gathering into a meaningful memory. To express our sincere thanks, I now invite Jayesh Aditya, Academic Subcommittee Member, IMA-MSN TNSB, to deliver the vote of thanks. Over to you, Jayesh.
Respected dignitaries, esteemed speaker, and dear participants, on behalf of Indian Medical Association, Medical Students Network, and Junior Doctors Network, Tamil Nadu State Branch, I take immense pleasure in proposing the vote of thanks for today's insightful state-level vaccination webinar series, IndiVax 26, on the topic Future Frontiers in Vaccinology. First and foremost, I extend our heartfelt gratitude to our esteemed speaker, Dr. J.D. Jayapriya ma'am, for sharing her valuable knowledge, insights, and experiences. Your session has truly enhanced our understanding and inspired us, ma'am. I would like to express our sincere thanks to the esteemed patrons of IMA-TNSB for your constant guidance, encouragement, and support in making this academic initiative possible. Our gratitude also goes to the state team IMA-MSN TNSB, whose leadership and coordination ensured the smooth conduction of this event. I would like also to acknowledge and thank Dr. Arunagiri sir, Dr. Ajay Karthik sir, Dr. Bala Hariharan sir, Dr. Hariram Vijay sir, Parthasarathi, Shruti, Avinash, Vishal, Aditi Balan, Harisha, and Jagrav for their tireless efforts behind the scenes in planning, coordination, and technical support. A big thank you to all of the participants for your enthusiastic involvement and active engagement, which made this session lively and meaningful. Lastly, we remain grateful to IMA for providing such a platform that fosters learning, collaboration, and professional growth among students and young doctors. With this, we conclude today's session. Thank you all once again for your presence and support. Have a great day.
Okay. Thank you.
Thank you, Jayesh. And with that, we come to close of today's session, session 4 of IndiVax 2026. On behalf of IMA-MSN TNSB and the entire organizing team, thank you all for joining us this evening and hope you have a wonderful evening ahead. Thank you, ma'am.
Okay.
Thank you, ma'am.
Thank you. Thank you for the opportunity and thank you.
Can I stop the line?
Yes, bro.